Genetic engineering explained. How genetic engineering changes DNA, and why the trade-offs matter. Genetic engineering is the deliberate editing of genetic material to change how an organism functions. In plain terms, it is the practice of rewriting DNA so cells make different proteins, follow different instructions, or stop following harmful ones. It matters now because biology has become programmable in a practical, repeatable way. Tools like CRISPR have made changes to DNA faster, cheaper, and more precise than older methods, which expands what researchers can test and what companies can build. But the central tension has not gone away. The most powerful genetic changes are often the hardest to control because living systems are messy, adaptive, and full of unintended interactions. This explainer will walk through what genetic engineering is, how it works at a mechanism level, the numbers that anchor it in reality, where it reliably succeeds, and where it breaks down in practice. The story turns on whether we can increase control faster than we increase capability. Key points. Genetic engineering changes DNA on purpose to alter traits, cell behavior, or biological outputs. Modern gene editing tools can target specific DNA sequences, but specific is not the same as predictable. Delivery is often the real bottleneck, getting the edit into the right cells, in the right amount, at the right time. Editing outcomes can vary cell to cell, even within the same tissue, which complicates safety and performance. Most real-world applications rely on careful trade-offs: efficiency versus precision, speed versus verification, reach versus risk. The difference between somatic editing and germline editing is not technical, it is ethical, legal, and generational. Misuse risk often comes less from evil genius scenarios and more from sloppy claims, weak oversight, and missing long-term follow-up. The field's next leaps will come from better control of outcomes, not just better cutting tools. What it is. Genetic engineering is the intentional modification of genetic material to produce a desired biological change. That change might be a new trait in a crop, a microbe that manufactures a chemical, or a therapy that alters cells inside a patient to treat disease. At its core, genetic engineering is about changing the sequence of DNA, changing how DNA is regulated, or adding genetic instructions that were not there before. The goal is to shift what the cell produces, or how the cell behaves. People often use genetic engineering as an umbrella term. Under that umbrella sit several distinct approaches: inserting new genes, often called transgenesis, rewriting existing DNA letters, gene editing, and changing how genes are turned on or off, gene regulation or epigenetic editing. What it is not is mindless Frankenstein tinkering. It is usually careful, incremental work that involves design, testing, verification, and repeated rounds of troubleshooting. What it is not, genetic engineering is not the same as selective breeding. Breeding reshuffles existing variation across generations. Genetic engineering can bypass that slow process by introducing targeted changes directly, sometimes in a single step. How it works. Genetic engineering works by connecting three things: a biological target, a molecular tool, and a way to deliver that tool to cells. First comes the target. You have to decide what you want to change and where that change lives in the genome. That might be a gene that encodes a protein, a regulatory region that controls when a gene is active, or a sequence you want to insert as a new instruction set. Next comes the tool. In gene editing, the tool often includes a targeting component that recognizes a specific DNA sequence and an effector component that makes a change. CRISPR systems are the best-known example. A guide sequence helps the system find the target DNA, and an associated enzyme can cut or otherwise modify it. Then comes the cell's own biology. When DNA is cut, cells try to repair it. Those repair pathways can be harnessed to disrupt a gene, introduce a specific change, or insert new DNA. This is where a lot of unpredictability enters, because repair outcomes depend on cell type, timing, and context. Finally comes delivery, which is frequently the hard part. You can have a perfect design on paper, but if you cannot get the editing system into the right cells, the result is academic. Delivery can happen using viral vectors, lipid nanoparticles, electroporation, or other methods, each with its own limits. A useful mental model is to think of editing as a controlled accident. You create a precise molecular event, like a cut or a chemical change, and then you rely on the cell to finish the job using its internal repair machinery. Your control depends on how well you can steer that finishing step. Numbers that matter. Efficiency is one of the most important numbers, even though it is often described loosely. Editing efficiency is the fraction of targeted cells that end up carrying the intended change. If efficiency is low, you may not get enough corrected cells to see a therapeutic effect, or you may end up with patchy traits in a plant or animal. If it is high, you still have to ask what else happened in the process. Specificity is another critical number, but it is not a single measurement. People talk about off-target effects as if they are one thing. In reality, off-target changes can include unintended edits at similar DNA sequences, unintended large rearrangements near the target, or broader stress responses that change cell behavior without changing DNA sequence at all. Increasing specificity can reduce some risks, but it can also reduce efficiency depending on the tool and context. Dose matters in genetic engineering in a way that is easy to underestimate. Delivery systems often behave nonlinearly: too little, and you get no effect; too much, and you increase immune reactions, toxicity, or chaotic editing outcomes. The practical problem is not simply more is better. It is enough in the right place, without excess everywhere else. Mosaicism is a reality check number, especially in embryos and early development. Mosaicism means not every cell ends up with the same genetic change, producing a patchwork organism. If you want a uniform outcome, mosaicism is a failure mode. If you are treating a disease in a tissue, mosaicism might be acceptable or even expected, but it complicates both safety and efficacy. Time is a number that shows up as kinetics, how long the editing tool remains active in the body or in cultured cells. A tool that persists longer may raise the chance of unintended outcomes, but a tool that is cleared too quickly may fail to reach enough cells. This is one reason transient delivery is often preferred, even when it is harder to engineer. Scale is a number that hits cost and reliability. Editing a small batch of cells for research is very different from manufacturing a consistent therapy at clinical grade, or producing engineered crops at agricultural scale. As scale increases, process variation becomes a first-class problem, where it works and where it breaks. Genetic engineering works best when the biological goal is clear, the target is well understood, and the system is forgiving. Many of the most successful applications fit this pattern. Microbes engineered to produce enzymes or materials, crops engineered for a specific resistance trait, and cell therapies where edited cells can be tested before being used. It also works well when you can select for success. In a lab setting, you can edit cells, test them, isolate the ones with the right change, and discard the rest. That ability to filter outcomes is a massive advantage, and it does not exist in the same way when you edit cells inside a living person. Genetic engineering tends to break when delivery is difficult, when small changes have big downstream effects, or when biology has hidden dependencies. Many diseases are not caused by a single broken gene, but by networks of regulation, inflammation, environment, and development. Editing one node in that network can help, do nothing, or sometimes make the system compensate in unexpected ways. It also breaks when the desired change is not just a DNA edit, but a long-term shift in a complex tissue. The immune system reacts. Cells divide. Edited cells compete with unedited cells. The body adapts, sometimes in ways that reduce the durability of the effect. A major practical limit is that precision in targeting a DNA sequence does not automatically translate into precision in outcomes. You can hit the right locus and still get a distribution of repair results across cells. That distribution is the true product you are making, and controlling it is hard. If the limits are mostly engineering maturity rather than physics, it is worth saying plainly, in many cases, the barrier is not that we cannot edit DNA. The barrier is that we cannot yet deliver, measure, verify, and control the full system well enough for routine, scalable use. Analysis. Scientific and engineering reality. Under the hood, genetic engineering is a competition between intention and biology. You bring an engineered tool to a cell and ask it to execute a change. The cell responds using pathways built for survival, not for your design goals. For many editing strategies, a key requirement is that the repair process follows the route you are counting on. If repair outcomes vary too widely, the same edit becomes many edits. That is why validation is not optional. You are not simply checking that cutting occurred. You are checking what the cell produced after it repaired the damage. Another reality is that measurement shapes belief. If you only look for small edits, you may miss larger rearrangements. If you only sample a subset of cells, you may underestimate rare outcomes. Claims can be weakened by more comprehensive sequencing, deeper tissue sampling, and longer follow-up. This is also where people confuse demos with deployment. A clean result in a controlled cell line or in a model organism is a demo. Deployment means messy genetics, diverse immune backgrounds, varied ages, comorbidities, and real-world logistics. Many "it works" headlines are actually "it worked" in a simplified context. Economic and market impact. If genetic engineering works reliably, the benefits concentrate in areas where biological manufacturing replaces chemical manufacturing, where agriculture reduces losses, and where medicine shifts from symptom management to causal interventions. Adoption depends less on the brilliance of the editing tool and more on boring infrastructure, consistent manufacturing, quality control, stable supply chains, and predictable regulatory pathways. The cost is not just the therapy or product. It is the monitoring, the training, the follow-up testing, and the systems that catch rare failures. Near-term pathways tend to favor applications where outcomes can be verified. Ex vivo editing, where cells are edited outside the body and then returned, often fits this pattern. Longer-term pathways involve in vivo editing, where delivery and safety become harder but the potential reach grows. Total cost of ownership shows up as maintenance burdens, long-term patient monitoring, repeat dosing if effects fade, and expensive testing to confirm edits are what they should be. In agriculture and industry, it shows up as compliance, containment, and reputational risk if trust is lost. Security, privacy, and misuse risks. The most plausible misuse risks are not always dramatic. They can be quiet and cumulative, overstated results, unregulated clinics, low-quality reagents, and rushed applications that skip long-term observation. Biosecurity concerns also exist in a more direct sense. Genetic engineering can lower barriers to manipulating microbes. That does not mean it is easy to create dangerous organisms, but it does increase the importance of oversight, screening, and responsible publication norms. Privacy is a subtler risk in medical contexts. Genetic engineering workflows often involve sequencing, variant interpretation, and long-term data collection. The more personalized the intervention, the more sensitive the data pipeline becomes, even if the edit itself is local to a tissue. Guardrails matter because incentives matter. Standards for measurement, independent auditing, and clear reporting of adverse events reduce the chance that hyper-competition drives fragile science into real bodies. Social and cultural impact. Genetic engineering changes how people think about disease and responsibility. If a condition becomes editable, social pressure can grow around who should fix what, who pays, and what counts as optional versus necessary. It also changes research practice by making causality easier to test. When you can perturb a gene or a regulatory element with intent, you can move from correlation to mechanism more often. That accelerates discovery, but it can also create false confidence if systems-level effects are ignored. Education and public understanding are shaped by metaphors. If genetic engineering is framed as editing a book, people may assume edits are clean, reversible, and perfectly understood. In reality, genomes behave more like living documents with overlapping annotations, context-sensitive meaning, and redundancy. Scaling access can empower some and squeeze others. The groups that benefit most are those who can afford advanced care, those near research centers, and industries that can invest in compliant manufacturing. Equity becomes an engineering problem and a policy problem at the same time. What most coverage misses. Most coverage focuses on the cutting tool, CRISPR, base editing, prime editing, and the race to improve precision. The overlooked constraint is that the editing tool is only half the product. The other half is delivery plus verification. Delivery is not a footnote. It determines which tissues are reachable, which cells are edited, how much exposure the immune system gets, and how controllable the dose-response curve will be. Many breakthroughs are really delivery breakthroughs wearing an editing headline. Verification is the quieter crisis. As edits become more complex, you need better ways to confirm what happened across many cells, not just in an average sense. The field's long-term credibility will depend on showing not only that edits can be made, but that outcomes can be measured comprehensively and explained clearly. Why this matters. The people most affected are patients with diseases driven by specific genetic changes, farmers and consumers navigating engineered crops, and industries shifting toward biological production. Researchers are also affected because genetic engineering changes what questions are feasible to test. In the short term, impacts show up as new therapies, new diagnostics, and tighter debates over regulation and safety. In the long term, impacts show up as a shift in how society defines normal, how risk is distributed, and how biology becomes part of the engineering economy. Milestones to watch are less about a single breakthrough and more about capability stacking. Watch for delivery methods that reliably target specific tissues, for standardized ways to detect rare unintended outcomes, and for long-term follow-up data that shows durability and safety in diverse populations. Real-world impact. A hospital lab edits a patient's immune cells outside the body so they can better recognize a cancer marker. The practical win is not only the edit itself, but the ability to test the edited cells before they are infused. A biotech company engineers microbes to produce a specialty ingredient that used to come from petrochemical processes. The consumer-facing product looks ordinary, but the supply chain becomes more resilient and less dependent on volatile inputs. A plant breeder uses genetic engineering to introduce resistance against a crop disease. Farmers see fewer losses, but seed systems, labeling norms, and trade rules become part of the success equation. A research group uses gene editing to switch genes on and off in a model system to map cause and effect in a disease pathway. Even if no therapy emerges immediately, the work changes what drug targets look credible. Frequently asked questions. What is genetic engineering in simple terms? Genetic engineering is changing DNA on purpose to alter how an organism works. The change might add a new function, remove a harmful function, or adjust how strongly a gene is expressed. It is used in medicine, agriculture, and industrial biology, often with careful testing and verification. Is genetic engineering the same as CRISPR? No. CRISPR is one method used in genetic engineering, specifically a powerful method for gene editing. Genetic engineering also includes older methods of inserting genes, newer approaches that change single DNA letters without cutting, and techniques that alter gene activity without changing DNA sequence. What is the difference between gene editing and GMO? GMO is a broad label for organisms whose genetic material has been changed using biotechnology. Gene editing is a specific set of techniques that can make targeted changes, sometimes without adding foreign DNA. Some gene-edited organisms may still be considered GMOs in certain regulatory systems, while others are treated differently depending on jurisdiction and the specific change. What are off-target effects in gene editing? Off-target effects are unintended changes that happen outside the intended target, or unintended outcomes near the target that differ from the plan. They matter because even rare changes can be important if they affect cell growth, immune recognition, or critical genes. Measuring off-target outcomes well is part of responsible development. Why is delivery so hard for genetic engineering therapies? Cells in the body are protected by barriers, membranes, immune defenses, and tissue architecture. A delivery method has to reach the right cells without spreading everywhere or triggering harmful immune reactions. Even when delivery reaches the tissue, it may not reach enough relevant cells to produce a meaningful clinical effect. What is the difference between somatic and germline genetic engineering? Somatic editing changes cells in an individual's body and is not intended to be inherited. Germline editing changes eggs, sperm, or embryos so the change can be passed to future generations. The ethical and policy stakes are much higher for germline editing because it affects people who cannot consent and can alter population genetics over time. Can genetic engineering be reversed? Sometimes an engineered effect can be mitigated, but true reversal is not guaranteed. If DNA is permanently changed in long-lived cells, reversing it would require another intervention that reliably targets the same cells. This is why safety and verification before broad use are treated as foundational rather than optional. What are the biggest real limits of genetic engineering today? The biggest limits are controlling outcomes across many cells, delivering edits safely to the right tissues, and proving long-term safety with strong measurement. In many applications, the gap is not the ability to edit DNA. It is the ability to do it predictably, at scale, with durable and verifiable results. The road ahead. Genetic engineering is moving from, can we change DNA, to can we control what that change means in a living system. The difference is the difference between a tool and a platform. One scenario is steady progress through better delivery and measurement. If we see reliable tissue-specific delivery with strong verification standards, It could lead to broader routine use in medicine and more predictable engineered biology in industry. A second scenario is uneven adoption, with success concentrated in applications that allow selection and testing. If we see ex vivo therapies and industrial microbes outperform in reliability and cost, it could lead to a two-speed world where some areas mature quickly while in vivo editing stays constrained. A third scenario is a trust shock. If we see high-profile failures tied to poor oversight or exaggerated claims, it could lead to tighter regulation, higher costs, and slower translation even for well-designed programs. A fourth scenario is a governance leap. If we see global alignment on measurement standards, long-term monitoring, and clear boundaries for high-stakes uses, it could lead to faster progress with less whiplash. What to watch next is not a single headline tool. Watch delivery, watch verification, and watch whether the field builds the boring infrastructure that turns powerful capability into reliable, accountable practice. More insight, less noise. Subscribe and explore the full range at taylortailored.co.uk.